Metabolomics and Microbiome, and Alison, Hamm, and Dan Manter from the USDA-ARS in Colorado will be giving us a summary on the microbiome project.
Hello, can everyone hear me?
Yes, we can.
All right. Let me pull this up. Okay, so today I'll be talking about the contribution and control of soil and co-microbiome on hop terroir and quality. This is a continuation of our projects. Just a brief introduction of why we even care about this. The plant and soil microbiome, which when we say microbiome includes all the microorganisms, we focus a lot on bacteria, but also fungi. It's really the health of the entire system of the plant, including the soil. And it influences the overall plant health. It's very generic, but there are specific instances. So it can help with disease, either directly attacking those disease agents or indirectly by improving plant resiliency. It aids in nutritional deficiencies by making nutrients, even if a lot of nutrients, even if you measure them available in the soil, they're not available to the plant in the form they are in. So microbes will do things like chelate metals for uptake. They also aid in abiotic stress, such as water, stress, temperature, salts, and other chemicals. And this year, especially, I would say that the hop plants were especially stressed by the weather. So far, our project accomplishments, like many other research this year has set us way back. But luckily, we actually sampled before we technically started this project back in 2019. So we have the samples, and it's just a matter of getting into the lab and getting them processed. So we did, again, just like in 2018, we did cascade and centennial plots. We also added on a really cool plot. Hopsteiner has they call it the museum, where it's their germplasm line. So these are 88 varieties, all within the same plot, meaning the same management practices. So we're gonna tease out the effect of plant genetics and each variety on its own host microbiomes. And then also we sent out a farm management questionnaire to each farm to see their inputs and try to determine some management practices that influence the microbiome. So as of today, which today it's actually changing very rapidly, we did get the kiln cone sequence that farmers sent from each plot that we sampled. We just got the farm soils sequenced around Christmas time, and we're still right now just looking at the data for the first time on that. So I'll have a lot of great information this summer to present. We still need to do the museum plot, send soil in for our cache analysis of nutrients and physical properties of the soil. We are awaiting on cone chemistry, oils and resins to look at the quality effects. And then we are also waiting on some more farm management questionnaires to be returned before we can compile that data. So these are some highlights from our previous year's research. We did find big differences in the soil microbiome, but both by state and within individual farms. We overlaid this PCOA plot with our soil data and found that climate is a strong predictor, as well as these other, you can see where the arrows are influences that are driving these differences. So our goals to add to this is we're going to add more farms, which would just make the data more robust. We're going to add management contributions, so say irrigation rates, fertilizer applications, and then also hop quality, so oils and resins. Doing the exact same analysis for our results this year, we found, again, clear separation between the 3 states and within farms. We have a lot more data to be compiled, and we're going to also combine the 2 to really get out the location differences. We also want to know the differences, the contribution of plant genetics, each cultivar, to the soil microbiome, which we only had 2, Cascade and Centennial, but they did separate out pretty nicely. But the location had a much more strong effect. Doing that again this year, we didn't see quite as much separation. We're not exactly sure why yet. We're going to look at this, obviously. But this is why we're excited about the museum plot with 88 varieties, because these are only comparing 2 and it's not statistically robust. So we were able to sequence some fresh cones. This is a completely new field for us. We really focus on soils, but there is a lot of literature out there in many crops indicating that the microbiome on the plant, in the phylosphere, is can be influential on, especially in disease and even in food quality. So we unfortunately were not able to detect bacteria or fungi in any kind of accurate manner. This isn't saying it's not there. It's just saying there's 2 reasons. It could be there's just very, very low abundance and we're not able to measure it with DNA sequencing technology. And also the primers that we use, the way that we set this up for sequencing, we could explore different primer sets and different methods to try to search for these bacteria and fungi. And we're working on developing those primers too. We also sequence kiln cones. So this is we wanted to know what are the brewers getting and what is the final outcome, because the microbiome on cones fresh is obviously going to change during the kilning process. We found even less bacteria on there, which is great for the brewers. You know, we would be concerned if it was overloaded with bacteria, because that could really affect the brewing process. Again, it's not saying it's not there. It's just in such low quantities, or our primer sets just aren't targeting that bacteria. The cones are extremely difficult because chlorophyll is, the DNA of chlorophyll is similar enough to the mitochondria of bacteria that also pops up and kind of overloads our sequencing results. So with that, I want to really thank especially the participating farms. You guys were out there with me digging and picking hops for hours during the day in the heat. Also, really want to thank the HRC for continuing this funding. We had some funding also from NRCS and USDA that we were able to supplement because we got the HRC grant. And also our collaborators, Hop Steiner working on our cone quality, and also Katie at CSU for all of her manual labor and support. So with that, I'll take any questions.
Okay, we do have a couple. The first one from Betsy, are you considering age of the yard, and do you think the source of plant material plays a role? Could certain nurseries have microbiome signature that ultimately carries to the yard?
Yes, so our plant age, we only chose mature plants between 3 and I believe 14 was the oldest yard we did. So we didn't measure babies. Since it is a perennial crop, we want to see, you know, planting a yard is going to disturb the microbiome. So we wanted to be representative. And yeah, You can carry over microbiome from a nursery to a field, absolutely. I think it would be really cool. Dan and I have talked a lot about how the microbiome should really be incorporated into genetic programs, because these, there are bacteria that are in symbiosis with these cultivars, and it's even possible, maybe, you inoculate the baby plants before getting into the field, and that microbiome will carry with them.
Okay, Alison, another question from Scott. How shelf stable will the crop year 2019 soil samples remain as they await lab analysis? Are there any concerns about microbiome drift while in storage?
There definitely are. And we store everything at -80 degrees Celsius, which is the best we can do. The soil, we did extract, we're able to extract the DNA and process those right after we got home in last fall. So, and the DNA itself is very, very, very stable. We do studies where the DNA is 30, 40 years old, and as long as it's stored properly. So we're confident that we are capturing the microbiome at collection as best as we could.
Okay, well, thank you, Alison. Appreciate that. And if you still have additional questions that you would like to submit, do that through the Q&A box, and Alison can answer those directly. So next up, under Metabolomics, will be Adam Heuberger, who is at Colorado State University in Fort Collins, Colorado. So Adam.
Great. Can you hear me okay?
Yes, we can.
All right, I'll go ahead and share screen. And can someone let me know they can see the presentation okay before we move on?
Good.
We're good? Great. All right, so my name is Adam Heuberger. I'm an associate professor of horticulture at Colorado State University. I'm here presenting on behalf of a research team at CSU. This is really led by Katie Nishaka, she's with us today, so she can help answer questions at the end of the presentation, who's with Odell Brewing, as a brewer and technician, and also a graduate student at CSU. So I'm really presenting a lot of her graduate research. And then also Harmony Bettenhausen, Jacqueline Chaparro, and Jessica Prenny, also at CSU, who are all working together to really explore the chemical makeup of hops and how we can measure that chemistry and understand how it affects quality. So what my laboratory does at CSU is I study what I call variation in plant and food chemistry. So if you take any kind of plant, such as hops, you can think of it as a pool of chemicals, which I'll also call metabolites. And we know that kind of pool of chemicals is driven by the genetics of the system, the environment, or where and how it's grown, and then also how it was processed. And depending on those factors, such as you have change in genetics, so you have different cultivar, will shift the chemistry of this. And so we say the chemistry varies based on these factors. The reason that we care about how this chemistry varies is because it can affect quality. So because it affects quality, we want to be able to track chemical variation. So if you shift the genetics, is that predicted to affect the chemical makeup of the hops? Or if you change management practices, how does that affect the chemistry or processing, for example? So in the chemistry lab, we really have two main approaches to analyze chemistry. The first is a quick and dirty approach. And this is really what's called predictive chemical or metabolite fingerprinting. So this is what you experience if you go to the airport and you get your hand swabbed. They take that swab, they run it through a machine, and that just takes a snapshot of the chemistry to be able to indicate whether or not a molecule is present. In this case would be, usually they're checking for TNT. So it's quick and dirty. There's not a lot of prep. It's just you get an instantaneous chemical fingerprint that says whether or not a molecule is there. On the complete flip side, there's slow and clean analysis, what we call comprehensive chemical analysis. And so this is what the term metabolomics is. So metabolomics is the deep description of the chemistry. It's a comprehensive profile of a sample. And there's value in studying, in this case, hops from both of these perspectives. But what I'll present today is our approach to doing quick and dirty analysis. And later, we're in the progress of doing our slow and clean or comprehensive metabolomics. So the goal of our metabolite fingerprinting method is to develop a rapid mass spectrometry method for hops. And this is because through fingerprinting and discrimination testing, we can assess several needs, such as, does this batch have a chemical profile that's expected for my program? So this is in lieu of having to smell or brew, but just getting a quick snapshot of the chemistry and saying, does this look like a fingerprint I wanted to have? Is my cultivar consistent? Can I trace the hops back to a specific origin? Am I optimizing agronomy and harvest for yield and quality? That is, if you have a chemical fingerprint that is ideal for your particular program. So the first thing that we need to do to develop a method to establish a chemical fingerprint is to determine the platform. And there's dozens, or honestly, probably hundreds of different approaches you can use. So we compared two what's called ambient mass spectrometry methods. So ambient means just out in the open. So it's a quick, uncontrolled analysis. And so the first thing we did was we explored what's called Reams. That's an acronym. I'm not going to spend much time on this because it worked, but it was not fun, and it was a little bit tedious. And so in this method, you're basically creating a vapor of a sample and then sucking up that vapor through a tube into a machine that collects a fingerprint of that vapor. What we're moving to now, and this is kind of the heart of what a lot of Katie is doing for research, is called direct analysis in real-time mass spec, or DART-MS. And this is a similar principle. It's ambient, so you essentially put your sample kind of in between these two pieces of the equipment, and then this piece of equipment right here is what establishes the chemical fingerprint for you. So it's instantaneous snapshot of the chemistry. So we did not like Reams, and so we're putting all of our attention towards DART-MS. And in the end, it's a very promising system. So the next step is what to analyze. And so these systems are developed so that you could take a hop pellet and wave it in front of the machine and establish a chemical fingerprint. But the data can be messy and a little less targeted than you want it to be. And so Katie explored all these different extraction methods and different solvent mixtures to be able to pull out metabolites to evaluate for the fingerprint. At the same time, we evaluated different authentic standards in the system to be able to see which terpenes and which bitter acids we know that we could actually detect using the system, some of which we were successful and some not. So we think that this instrument has the potential for direct analysis of quality-related metabolites in hops. The other kind of major decision we have to make is when running mass spec as a method is what's called positive or negative mode. And this is to actually make a measurement. You have a mixture of metabolites in your extract. You have to either add a positive or negative charge. You can't measure neutral or metabolites with neutral charges. So it has to be one or the other. And if we add a positive charge, we compared that data versus adding the negative. And in the end, the negative was a little bit cleaner and more sparse, and the positive was a little bit messier with a lot more noise. And in the future, I'll explain kind of we're moving forward with a negative. It gives a really nice, clean, simple signal to analyze. The next is to further optimize the platform, and something that we experienced. Not only do we experience a pandemic and have the challenge of trying to manage research on campus with all kinds of new restrictions, but we had the state's largest wildfire in Colorado history happening in our front door. And so this is what our atmosphere looked like while Katie was trying to collect data. And given this is an ambient method, you were seeing a lot of noise, particles in the atmosphere show up in the data. And so she went back and worked with the machine shop at Odell Brewing to help craft basically different ways of kind of protecting the sample and kind of cutting down on this noise when collecting the fingerprint to give a cleaner signal. Another thing that we're working on to try to optimize and kind of reduce noise in the data, because this is an ambient method, is what's called pulse gas technology. I'm not going to spend time getting into the details of this right now, but I'll just show you in the end, we have these really beautiful, clean fingerprints of different chemistry. In this case, these are the fingerprints that correspond to Cascade, Centennial, and Strata. And they look similar because they're all hops, but they're slightly different. And that's really what we're kind of going after, is what are these chemical signatures that differentiate these hops and define them is related, that's important to quality. So kind of the last step where we're at right now is optimizing the statistics. And so this is, once we collect these fingerprints, how do we analyze them? And so in this case, we have to figure out what math we're applying to associate a specific fingerprint with a cultivar, an environment, or a sensory characteristic. And so this I have here just listed a number of different parameters that we're exploring once we actually collect these fingerprints, really getting into the weeds of how we apply math to the data to tell us what we want to know from this fingerprint. So the overall workflow is how this happens is we collect this fingerprints of knowns from our 3 different samples. We actually create models from the knowns of saying, okay, what are the different signals in these fingerprints that really define a centennial or a strata? And then, this is where we're trying to go with this, is then we can take unknowns. So we can take 3 hop samples where you don't know what variety they are or what farm they came from, and evaluate the fingerprint and try to determine which one it looks most similar to. So if you're in a breeding program or if you're applying different agronomy and you suspect that it might shift the overall quality or the chemical profile of that hops, you can, with this fingerprinting method, essentially assign it to specific categories in an instantaneous manner. So that's kind of to conclude where we are with this particular research area. We identified best extraction method, so we settled on 80% ethanol, a really good MS platform called DART-MS, and then various model parameters that can differentiate samples based on chemical fingerprints. And so what we're looking for is, it looks like hops, but we can see slight differences depending on cultivars. And as we move toward, we're going to see if we can evaluate environments as well. What we're still doing is still tweaking the fingerprint acquisitions. Background noise is still a problem, but there's little tweaks we can do to try to really optimize the system for hops. So the next steps then is to kind of associate this data and take these fingerprints and see what we can really do with it. So for example, here's some sensory plots that Katie had done. For example, on Cascade, she took these samples from different farms. You can see from farm D, this has a slightly different sensory profile. So now we want to dig into these chemical fingerprints and say, is there a signal in this fingerprint that would indicate that this would have a different sensory profile than the others? The other is to be able to apply this information to a quality control or quality assurance setting, especially if you're doing different processing or if you're concerned about where your hops are actually coming from, to be able to say, does this fingerprint look like it should? And then continue to evaluate different platforms as well. So very briefly, for the sake of time, I'm going to talk about the flip side, which is instead of that quick and dirty, what we can do with these fingerprints is how deep can we go to really understand the chemistry of hops. And so this is our current really active research area, is to perform deep chemical profiling to better define hop chemistry in its relation to varietal differences, effects of agronomy, and flavor. And the rationale behind this is, while fingerprints tell us if chemical variation happened, right? They can give us a signature and say, this hop isn't exactly how we expect it to be. They don't really tell us anything about the links between hops chemistry and quality. So they don't tell us what chemicals we're seeing and why it's driving specific differences in quality. And so this is why we're working towards metabolomics, the study of all chemicals in the system. It'll inform on chemicals, so we're looking at metabolites or small molecules, also metals. We have that expertise as well, but we're trying to figure out what's essentially driving quality, and then what is basically how that quality is shifting based on genetics, environment, and processing conditions. So our experimental design is that we have 18 unique hop samples. This is what Katie had spent the last year running around collecting all these samples, I guess flying around. So there were 3 cultivars that were working with Cascade, Centennial, and Strata, from 3 hop farms, secret locations on their request, and from 2 different states. We're running all these different chemical platforms. So we're doing a really intense extraction method to pull out all the metabolites we possibly can. We're running it on, these are 4 different mass spec platforms. And this one we're running twice in both positive and negative mode. We're doing volatile analysis and metals analysis as well. Like I said, we're going deep. Katie's been performing sensory analysis on these as well, which we're going to integrate the chemical and the sensory data. So what Katie has been finding is that it is a challenge to do sensory during the pandemic, but she has successfully been able to do quantitative descriptive analysis, so using a scale of 1 to 5, which within really common categories like citrus and tropical, on these 18 samples that are from, different cultivars from different places. And the idea is that we kind of want to see small differences in sensory variation that we can then link to the small differences in chemical variation at the same time. So where we're at is we're in the process of running the metabolomics. And so the type of data that we expect to see, we expect to get information on about 5,000 compounds, and we can probably annotate about 1,000 of them. So that's pretty good. The types of compounds I'm talking about are things like phenolics, amines, amino acids, alkaloids, terpenes, non-terpene aromatics, lipids, sulfur compounds, and many, many others. So we're trying to go very broad in the type of chemistry that we're sampling. We expect to see about 30 plus metals out of all these samples as well, and then to be able to quantify and look at the effects of these genetics and environment on the different chemistry. And then specifically pull out metabolites that are known to be quality related, such as terpenes and organosulfurs. So just a quick summary, summarize this presentation. Through fingerprinting, we can basically establish these chemical fingerprints and differentiate hops, which we think is important for this true to type analysis. The negative mode acquisition, the one thing I forgot to mention right here is that it's excellent for this bitter acid detection. And so this could be a really nice way of measuring those compounds instantaneously. And that, I think we would have to go a little bit deeper and see what is the level that we can accurately quantify those types of compounds, as well as some terpenes we could see as well. And then through the metabolomics, we'll be able to fully describe the chemistry of hops, to differentiate cultivars, predict effects of environments, and ultimately link that metabolite data to differences in sensory. So I already mentioned this is a team effort with the authors. These are non-authors that are also part of the team. Folks from Colorado State University, Dr. Paul Matthews and Dr. Patty Arone, who were helping us kind of design our metabolomics experiment, and Scott Dorsch has been a huge help all along the way. And to acknowledge our funding from the Hops Research Council, and then all the resources that Odell has been providing to this as well have just been huge in getting this to happen. So this time I'd like to thank you in the audience for your attention, and I'll see if there are questions to answer.
Adam, we do have a question from Mark. He asked, how many compounds are you up to relative to your defined fingerprint compounds like mycene so far?
So things like our quality compounds, I want to say we're probably solid on 5 or 6 known quality compounds. And that was by running standards and kind of matching signals. The challenge with the fingerprinting is we have no way to validate that. So the thing that we can do is kind of work with different hops and spike in compounds and really validate what we're seeing. The other nice thing is once you know that a compound is there, like myrcene, you can fine tune your model. If that's something you really care about in your program, you can fine tune your model to put more weight onto that of saying, oh, in my processing or in my agronomy, did I shift this, and then basically have the model kind of focus on those types of quality compounds. So likely in our metabolomics analysis, we expect to be in the hundreds of compounds that we know are important to hop quality. Just thinking about the number of compounds that are relevant to sensory and fermentation processes.
Okay, Adam, could you give us just a quick timeline for the basic fingerprinting? How long does that take?
In terms of collecting data, Katie, if you're here, do you want to speak towards what it's like to actually collect the data?
Yes, absolutely. Can you guys hear me?
Yes, we can.
Yes.
Great. So it's instantaneous. So it just depends on how large your set is. And I included quite a few technical reps. So it takes me about a day to do all those different samples, 12 unique hop samples, 3 replicates. I wasn't able to get biological reps because actually the hop growers that were participating sent me all the samples to make it easy on them. They just sent me brewer's cuts, but it's right away and you get the spectra right away. And then the modeling just takes a little bit of time, other back end to create those models.
Okay, thank you. Okay, well, thank you very much. Very exciting work, and we look forward to continuing this project.
Great, thank you.
And if there are additional questions, please respond or ask your question in the Q&A box, and Adam can respond to this directly later.